Hello, everyone. My name is Roger Hajjar. I'm a cardiologist specializing in heart failure and cardiac transplant. My home institution is Mass General Brigham in Boston, Massachusetts, where I direct the Gene and Cell Therapy Institute across Mass General and Brigham hospitals. Today, what I'd like to discuss is dilated cardiomyopathy secondary to a specific mutation called phospholamban R14 deletion. So, what are the unique consideration about this cardiomyopathy? And that is, it starts with how the contraction of the cardiac cell occurs on a beat-to-beat basis. And this happens when the membrane of the cardiac cell becomes depolarized when an action potential comes through. Channels open up, allowing calcium to come in, and that small amount of calcium induces the release of a higher amount of calcium from an organelle within the cardiac cell called the sarcoplasmic reticulum. That large amount of calcium that goes into the cell then activates specialized proteins called myofilaments, which allows the contraction to happen. That's when the force develops, that's when the heart contracts. And during relaxation, calcium is then taken back up into that organelle called the sarcoplasmic reticulum under the guidance of a specific pump called the SR calcium ATPase pump. And that pump is quite important because it regulates the entry of calcium out of the sarcoplasmic reticulum as well as its entry inside the sarcoplasmic reticulum. Now, there is a small protein called phospholamban, and that's really the target gene that we're going to be discussing. And that protein is regulates the sarcoplasmic calcium ATPase pump, also known as SERCA2A, because it puts a brake on that pump when it's not phosphorylated. And when it gets phosphorylated, that pump activity increases, so the brake is released and you get more activity. So for example, when you run and you have a lot of catecholamines, which are these specific hormones that allows your heart to increase its rate of action and relaxation, phospholamban gets phosphorylated and SERCA2A is allowed to pump more calcium back into the sarcoplasmic reticulum, allowing it to release more. So you have an increase in the amount of force and amount of contraction that can happen. So phospholamban plays a very critical role in the sort of day-to-day activity of the human body. Now, the phospholamban gene is on a specific chromosome called chromosome 6. And just like any other protein, it's made up of small building blocks called amino acids. And those amino acids in series allow the protein to function in an exact way. So for phospholamban to put the brakes on SERCA2A when it needs to, and when it gets phosphorylated at a specific site on one of these building blocks, it basically that brake is released and you have increase in activity. Now, phospholamban has been known to have mutations in these building blocks, meaning that instead of a specific amino acid being there, another one is there because the genetic information coming down is altered. So early on, there were a number of mutations that were identified, and we call them mutations by the site, the number. So if we say there's a mutation at R9C, that means that at position 9, there was a shift from one specific amino acid sequence to another. If we say there's a mutation at L39 stop, it means that at position 39, the amino acid is mutated and the rest of the amino acids are deleted. Now, there's one specific mutation called the R14 mutation, which is a deletion of that amino acid, and it occurs at position 14. And at that position, R, which represents arginine as the amino acid, is deleted. And that deletion causes a specific kind of cardiomyopathy that is dilated, but also has very significant arrhythmias. This mutation was first discovered in a large Greek family with hereditary form of dilated cardiomyopathy in 2006. And that family was found to have a heterozygous form of R14 deletion, meaning that there is one copy of the gene that makes normal phospholamban and another copy that makes the mutated form of phospholamban known as R14. And these patients, the first time that this was discovered, had very severe dilated cardiomyopathy, meaning both their ventricles, the left and right, were dilated and had very poor contraction and were in severe heart failure. And many of them had perished or had cardiac transplantations. Now, in 2012, this mutation was also identified in the Netherlands. And in the Netherlands, there was found to be a much larger cohort of patients as of last count, over 2,000 patients carrying this specific mutation. And these patients in the Netherlands were also found to have high degree of arrhythmias that comes from the left ventricle or the right ventricle, and also were afflicted with ventricular dilatation and dilated cardiomyopathy. After a lot of studies, this mutation in the Netherlands was actually pinpointed to a specific area in the north of the Netherlands, and that's known in Groningen. And it's thought that 500-600 years ago, a family emerged from there with this mutation, but they were able to survive and procreate and have kids who unfortunately continue to have this mutation. And these patients present at different times in their lives with either early forms of the disease, which can be just having rhythm problems that's picked up on an electrocardiogram, all the way to severe ventricular arrhythmias that could be deadly, and of course, end-stage heart failure. So there's a variation of the disease that can manifest itself from very mild to extremely severe with deadly consequences. Now, the R14 deletion mutation has also been found in cohorts of patients in North America and also South America. In the US, for example, there are now maybe 50 to 60 patients who've been identified with this mutation, and as more and more testing is being done that's targeted for this mutation, we're finding more and more of these patients who have this mutation. So the number appears to be constantly growing, and because of that, we have more awareness about the disease and how it manifests and how it can basically advance with age. So as I mentioned, many of the patients who present for the very first time can be at the very end stage. And for example, in the Netherlands, a high percentage of patients who present for the first time with heart failure have this mutation. So usually that means that they've been carriers, they've had early stages of disease, were not detected, and then now they are at the end stage in their third, fourth, or fifth decades of life. And the penetrance, as we call it, so how fast the gene expresses itself and how fast the diseases manifest is still unknown, and it's variable from one patient to the other. So that's why in the Netherlands, there's been a very strong focus on identifying patients early on or identifying carriers early on. And the Phospholamban Foundation, which started in the Netherlands and now is in the US and other countries, has been focusing on trying to test as many of carriers as possible by doing widespread testing in different cohorts of the population. So that's a big endeavor that's ongoing right now with these phospholamban foundations. The foundations also work very closely with the clinics, the heart failure clinics, and the cardiomyopathy clinics across Europe and also in the US to make sure that this mutation is added as part of the different types of testing that occurs when patient presents with unexplained type of congestive heart failure or unexplained type of arrhythmias early on. Now, digging deeper into what really happens when this disease really settles in within the heart, there's a very specific type of pathological manifestation when you look under the microscope and you see loss of cardiac cells when the disease has progressed to a very late stage. And then these lipid droplets that seem to occur across the muscle of the heart. And that's where the cause of the arrhythmias comes from, and that's where the dysfunction and the contraction is occurring. So as you can imagine, there's been many studies that are very much focused on understanding how this disease is progressing. In terms of the patients, as the diagnosis is made, patients are placed on different types of heart failure medications, and that can be geared towards the arrhythmias that they're experiencing. So many of these patients receive intracardiac defibrillators so that in case they get arrhythmias, they can be shocked out of it. Many of these patients also are on medications that are used for congestive heart failure, for the treatment of congestive heart failure, with variable responses. Sometimes they respond appropriately, sometimes the medications don't work as well because the genetic mutation can really cause specific damages that are not responsive to medications. The ultimate treatment at this time is cardiac transplantation when the disease is very severe and the patients are in late stages of heart failure. And obviously, this is a type of treatment, the cardiac transplantation, that can happen to only a few subsets of patients who need it. So clearly, there is an unmet need for this medical condition, for this cardiac condition. And a lot of efforts have been focused on using genetic types of therapies to treat the disease, and also small molecule RNA interference and RNA therapeutics have been deployed to basically treat the disease. And one of the big efforts that has been successful is to have models for this disease, both in human tissue models, whereby tissues from different patients can be taken and then hearts are grown from those tissues, which manifests the disease the same way that the patient has. Also, there are multiple animal models, both in rodents and pigs, that have been developed with this mutation so that testing can be done. And what are some of the novel therapies that are being considered? So one is a gene therapy approach whereby you try to enhance the phosphorylation of phospholamban that's available. So that's a normal phospholamban. These patients have one normal and one disease, and you try to target the one that is not disease and enhance its activity to overtake the non-functioning one. And that's being done by a gene therapy approach. There are ways to correct that mutation. So the R14 is mutated. So can you correct it by adding the right gene sequence? And that can be done with a technique called CRISPR or gene editing. And that is happening right now in experimental models with some reasonable success. Another way of addressing the problem is to basically target the disease gene that's causing mutation and bring down the level of RNA expression. And that's what we call interference RNA or short-haired RNA that's very specific to that disease gene. And that's also part of the armamentarium that we're deploying against this disease. There's small molecules that target the disease gene to shut it down or to target the diseased protein, the phospholamban mutation, that's also being tested. So overall, we have a genetic disease, the R14 deletion, that started out with one single family 20 years ago in Greece, and now has been shown to be quite prevalent across not just the Netherlands, where it has a very big percentage, but also across the world. And this has been thanks to the awareness that has been increased over the years. As a field, in terms of the focusing on phospholamban, investigators have been put together from different varieties of places, from epidemiology to genetic engineers to pathophysiology experts, and have come together to really define where the targets should be for this disease. And as one of the investigators who's been fortunate enough to be part of this community, I feel very bullish that this is a mutation that has specific therapies coming down the line, whether it's genetic manipulation or small molecule manipulation or RNA manipulation, that will revert the mutation back to normal, and we should end up with a normalized heart in the next 5 to 10 years. Thank you.
This video is part of a series of educational videos developed by the Genetic Cardiomyopathy Awareness Consortium and brought to you by the DCM Foundation. Our goal is to connect you with the leading experts in the field of cardiomyopathy to further your understanding of this disease and to provide resources to help you manage your care. Thanks for watching.